Hey everyone, and welcome back to Pipetting Hot Tea, the podcast where we share stories, insights, and discoveries shaping the world of science. We're your hosts, Anusha
and Amelia.
And today's episode is a deep dive into the microscopic world of structural biology, where the architecture of molecules tells us a story about life, disease, and potential cures.
Today we're joined by François Couture, a full professor and chair in the Department of Biochemistry, Microbiology, and Immunology at the University of Ottawa. Dr. Couture is a protein crystallographer by training and an expert in structural biology and epigenetics, with more than 90 scientific papers to his name, some in major journals like Science, Genes and Development, and Cell. Dr. Couture and his team are working to uncover how tiny changes at the molecular level can lead to big breakthroughs in treating diseases like leukemia.
So how did a fascination with molecular structure lead to research that might one day help fight cancer? That's exactly what we're about to explore. So let's welcome Dr. Couture.
So how did you get into science, and what led you specifically towards biochemistry, immunology, and microbiology?
I will try to provide an answer that won't take 2 hours, but I would say that as a student, I was always curious, and it started really early on when I was not even an undergrad, but high school, and I was also what I consider myself an artist. So I was singing, I was playing instruments, oboe, if you ask. I was also in a play. I like improv. I was really into all these sort of things, but also like academics. I really like math. I really like biology. I really liked chemistry. So for me, at a point in time when in Quebec, we have to pick the cégep, right? And I had a chance either to pick the cégep to go study in science, math, physics, chemistry, and biology, or choose to go to Saint-Hyacinthe, which is where they have a college for those who are interested in acting and these kind of things. And I decided to go and stick to science, but continue to do some acting, some music playing on the side because I really liked it. And then when I start cégep, this is where really things start clicking a little bit. And it started actually in my second year, I would say in the third year, because I didn't know what to do. And typically a cégep, for those who wants to go to university, it's 2 years. For me, I decided to do it in 3 years. Not because I'm lazy, it's just because I didn't know what to do. I was interested in science, but I was still interested in arts.
Yeah, so it must have been a difficult choice to choose between
those
two
things. It was. But when I realized at some point, I'll get to that when I started taking courses where you had a questions linked to it, a lab portion that actually challenged you to have ideas as to how to solve this problem, this is when things started clicking a lot, because now you had a creative process embedded into a scientific question, which to me was kind of the perfect marriage of my interest, which is a creative process built within a scientific question. So to me, it's like, oh, that's really interesting. And in the third year in Cégep, I decided to do an extra org chem course. Who does that, right, in Cégep? And essentially, the additional organic chemistry course was actually a special project. There were no defined boundaries. You could do whatever you want. So essentially, in discussion with Sherbrooke University at the time, we decided to embark on the synthesis of a neuropeptide. And we had to look at the protocols, buy the consumables, troubleshoot, and really come up with the idea of how we would synthesize that peptide. So to me, again, we were achieving something, producing something, yet troubleshooting within an area that there was a creative process behind it. So that was really something that stimulated me to go to university and say, okay, I'm into research, but I don't really know what I like. To be quite honest, at the university, I liked everything, right? And I think talking to science students, you're all like that. You all find everything interesting. So essentially, when I started undergrad, which was very overwhelming, a lot of students, you kind of get lost into your, sometimes for me, I got lost in my initial passion a little bit. It's like, okay, I like creative process and I like labs, but the first year there was not many rooms to explore that, to do that. Until my second year at the university, during which between the second and the third year, or at the end of the second year, you can work, it's part of your course curriculum if you want to do it, you can, for course units, spend the whole summer in the lab. So in the form of a 6-course unit, I could actually work the whole summer in the lab, which really, where I literally fell in love with science. Because I was in a real lab doing real science and really exploring my creative, or really exploiting my creative profile, so to speak, into science. So I really liked it. And to be in a lab environment, not for academic purposes, but really to save lives, really changed the whole dynamics of the way I approach science. And to me, that was really a perfect match. On top of that, the lab where I was, the lab of Dr. Breton at Laval University, was actually using a technique called X-ray crystallography to elucidate protein structure in 3D. So I was kind of, as part of my project, I was involved in building protein structure in 3D, which sometimes I consider, or analogous to a piece of art. This is a 3D structure, you're building something, and out of the structure, then you can hypothesize what would be the role of this particular amino acid, for example. What is the specific role of this enzyme? What is the specific impact of this particular mutation on this enzyme, and is it involved in some sort of disease progression or some sort of disease? So to me, all this together really made the whole thing click. I like science, I like the creative process, and then that, protein structure determination is pure. You cannot have more pure biochemistry than that, or really basic biochemistry. This is basic understanding of the role of an interaction that you will see between two residues within a protein, for example. So to me, really, this is how my journey led me where I am today, really.
That's such an amazing journey. So can you define what structural biology and epigenetics is for us, and what part about it excites you the most?
Yeah. Structural biology first. I see the building. Why? Because I use my eyes, and the visible light oscillates in some sort of range of light that I can see the details, right? But at some point, you get to a point that things are so small that you can't see them with your naked eyes, right? We're going to talk about an organelle. If you don't have a microscope, you're talking about a virus, a bacteria. You can't see them with the naked eye. Imagine trying to look at a protein structure that is several magnitude of order smaller than what I just named. How you go about it, right? So how you try to see what is the three-dimensional organization of the amino acids forming a protein that carry out some sort of biological function. And for that, in my lab and in many other labs in the world, we use what we call X-ray crystallography, which is a technique using X-rays that will expose a protein sample to X-rays, and the way light interacts with the protein will give us an image of the protein three-dimensional structure. What excites me the most about structural biology is, again, it's building and understanding how the protein, all these residues come together in 3D, right? If you think about a primary amino acid sequence, this is relatively simple, semantic, or letters one after the other. But evolution has made that all these letters corresponding to each one residue comes together in 3D and performs some sort of biological function, right? And when you get an information, what excites me the most is that you can tease out several type of information, but the three main ones that I'm focusing on is are there mutations that have been sequenced or identified in patients suffering from a dreadful disease that are actually perhaps impacting the protein function. Now that I know this protein structure and the relative orientation of all these residues, then I can understand better what could be potentially happening to the protein when that mutation is deposited or appear in this particular gene on this particular patient. And now more than ever, we can extract that information and design therapeutics to buffer the impact of that mutation, for example. The other element that is really, makes me wake up in the morning and go to the lab, it's really about developing inhibitors. When you get the three-dimensional structure, then you can have a better idea of the regions that are important for the enzyme to carry out its function. So using in silico methods, and we do that in our lab, we can design in silico using the structure of the protein, and then we can place in silico all sort of chemical scaffolds and figure out which one is the best. And actually, this is how pharmaceutical companies start designing inhibitors targeting one particular enzyme that they think contribute to the progression of cancer. So that's another thing that is really, really, really nice. And the third thing's very much fundamental is just sometimes when you solve a structure, you're the first one to solve the structure ever. We don't repeat the work that has been deposited or published elsewhere, right? So just to get an insight, the first at how this protein functions three-dimensionally and how the arrangement triggers its function is quite amazing in my opinion. So that's structural biology. Epigenetics. Epigenetics, how can I say this? We have in our genome pretty much the same, we share the same sequences, right? And so yeah, we have some SNPs, some variation based on population, things like that. You have variation. Yet, you would take, for example, two identical twins, okay? And they would be separated. There are examples of that at birth, right? And one will live in, would say, very wealthy, comfortable, well taken care of environment. The other one will not have the same chance. The same individuals at 20-year-old down the road will likely be different in terms of genetic signatures, traits, and these kind of things. Some things are similar, but some things are different. And one aspect controlling this is called epigenetics. Epigenetics can be, to make a somewhat of a comparison, is, you all know Margaret Atwood, I guess, a famous Canadian writer. So a writer will write a story with words and letters, okay? And the editor comes in and says, you should change the word. You should remove words. And sometimes that may change the story that is being described by the author. Epigenetics is the same. However, the book is DNA. So epigenetic mechanisms will be there to actually hide or expose DNA sequences that will have to be, for example, exposed DNA sequences that need to be interpreted by our transcriptional machinery, by our replication machinery, by our DNA repair machinery. And sometimes these epigenetic processes will hide the DNA. So it's like the editor removing words. The epigenetic mechanisms are there to hide. The sequence is still there, but is not being interpreted or read by our nuclear machinery. Going back to the example of the two twins, during their exposure to different environment, there are likely epigenetic processes that took place that give some traits to one individual over the other. This is one example. There are millions of examples out there. For example, beautiful stories or studies that have been publishing in Science, for example, about bees. You know, you have the queen and you have different roles for the bees, right? But once the queen dies, what decides that will make the new queen? Well, actually, it is now known that epigenetic mechanisms come into play and that actually come from the honey, and some hormones that they're producing will change the chromatin environment, the DNA environment, and will start expressing genes or repress some genes to make sure that the next queen or changing roles into, I call this the honey, the whatever, the environment, right? So that's another example, and there's tons of that out there. But yeah, epigenetic processes, if you, in a nutshell, it's our mechanisms that are there to expose or hide DNA sequences that will change the behavior of a cell to an environment, to a particular environment, and more at the macroscopic level, or I would say, I see you there, then different threats, right? Yeah.
So then how does structural biology and epigenetics kind of relate back to the work that you're doing right now?
I would say that structural biology relate to any kind of biological science that you do, I'll be honest. So obviously in epigenetic research, we want to understand how, for example, when the DNA sequence is not fully opened up, what are the mechanisms that help open up these regions, right? So my lab works on different regions of proteins that will, using different mode of energy, so for example, ATP consumption, so for example, they will open up chromatin and they will expose that DNA sequence for a different kind of activity. Okay. Well, this initial mode of engagement of these machineries onto our DNA takes different form. And sometimes we're going to find a region of a protein that really latches on something on DNA or protein-DNA complexes that will change its conformation. So when it comes very handy for structural biology is to understand how this enzyme, for example, how this new region on the protein engage into that interaction and do its job.
Okay, so it kind of lets you like 3D map out what the new interaction is going to look at?
Exactly.
Your work is also, we saw that was related to like leukemia. We saw that there.
Yeah.
So how does the structure of biomolecules influence diseases like leukemia?
Yeah, that's a good question. So my lab, when I started my lab, I started working on this complex called myeloid lymphoma leukemia, or MLL, protein complexes. It's a large family of enzymes heavily implicated in pediatric leukemia, some adult leukemias as well. And the way we go about it is we know that certain regions of that complex are important for its activity. So for us, when we started delving into this, we said, well, if we understand how the protein complex assemble using structural biology, for example, and we can relate back to what I said earlier, one of the things that excites me about my work is we understand the 3D structure of some of these proteins. We can design small molecules inhibitors preventing, the assembly of that complex. So essentially we made some progression on that front. And we've been collaborating with many groups in the world, and some of them actually managed to define a new molecule or to devise a new molecule that is actually currently used in therapeutics. So they're good. So there's one aspect of the research. The other aspect of the research is that there's several aggressive mutations, or not I would say aggressive mutations, but mutations that have been associated with aggressive forms of leukemia. And for us, this is where our work tangent more, I would say, trying to understand why these mutations trigger such aggressive form of leukemia. And at such young age, actually, we've had dealt with mutations identified in infant or baby that are 3 months old, right? So unfortunately for this patient hasn't been exposed to much environmental stress. You think about this compared to an adult, for example, and yet carry this mutation, which triggers really very unfortunate consequences. Yep.
Yeah, so kind of coming back to that, is there a way that kind of epigenetics could kind of come and be related to that kind of research?
Yeah, absolutely. And there's so many examples out there, and it's just coming together, I would say. If you look, you always have success stories that go really quickly. The vaccine for COVID is one of them that goes from really, I would say basic discovery to translational and, application in human. But in epigenetics, similar to other fields, for example, there's been a natural progression in terms of, finding drugs that will target what we call the epigenome. What I call epigenome is the ensemble of mechanisms that control epigenetic signaling. And there are some success stories out there that there are some treatment that have been approved treating some cancers that are target epigenetic enzymes. So for example, histone deacetylases. We have some histone methyltransferases now well-known targets. So it's just the beginning. The last 5-10 years, we've just seen a really steady but increasing, increasing number of publications on the development of, primary inhibitors, maybe not good for the clinic yet, but, published. So it means that really the pharma is right behind it, and it's just, it's coming together. So absolutely.
Okay. Yeah, so I mean, in your research, what are some challenges that you go through and how do you work through them?
Many.
Many?
The most, so I think that, I think that I see challenges sometimes as opportunities to get better, right? So I think there was few studies in Canada in published out there a few years ago that were showing that in Canada, we're producing very good science with the level of funding we have, right? But the competition is fierce. So obviously one of the challenges is really getting the funding to do the research, and there's so many mouths to feed, and I understand that. So it's very competitive, and also so you have to go every 6 months. You need to go out there and ask for more money, come up with innovative project, address reviewers' concerns. This is a very stringent process. And this is a good thing, right? Because you want to make sure that at the end of the projects that are funded are doing the best science in the world, right? So this is one challenge, but it means that you just need to get better, read better, network, right? Establish connections with collaborators that could address one of your weaknesses that were out, for example, one of your weaknesses as a PI. Because one thing that is really cool about my job is that I learn every single day. I don't go to work, I go to school every day. If you think about it, because I learn something new every day, which is an exciting part. But I realistically, I cannot learn everything. So there are people better than me in certain areas. So one challenge brings an opportunity to actually establish a strong network that surrounds you to really get better. Another challenge is to recruit good students. Believe it or not, we pay the students, they have a good stipend, and obviously you're not making millions of dollars, but recruiting students that are legitimately interested in research, and it's hard these days. The students are more informed, which is great for you guys, because when I was an undergrad, I was not as informed as you at all. So for me, it was med school or research. I was not seeing any kind of other options. And there are tons out there now, we know. But again, another challenge for me is really recruiting good students. But on the other hand, that brings an opportunity to get better at reaching out, doing outreach activities, to send emails, to prepare a better website, to really be on social media, these kind of things, so to be a little more proactive. I started my career in 2007. Social media is still something that I learn every day, right? So again, challenges bring opportunities. And finally, try to wrap up a normal work day in less than 10 hours. It is a challenge. For me, and for many researchers, this is not a job. This is a passion. Right? And so to establish kind of a work-life balance, very challenging, because for me, again, I don't go to work, I go to school, I go to learn, and I would spend all the hours that I could if I was alone in the lab, because what I like to do is do experiments, learn in the bench, and trying and find new things. Right? But the reality is that I have kids to take care of, and a wife that would like to see his husband showing up once in a while at home to help to cook the dinner, right? So again, the challenges bring opportunities for me to better reflect, to balance things out and, seek opportunities. And they're great support at the university here for prof to really strike some sort of balance in your life. But that's another challenge.
Yeah, so you mentioned having to work like 10 hours like a day. So can you walk us through a little bit what a typical day kind of looks like for you?
That's the most fun, that's the most interesting, not fun, quick question that I get because I cannot give you an answer because not a typical day. Seriously, every single day is different. So for example, today I troubleshot two things with my students. I read a paper, I had few meetings. Tomorrow, I don't have meetings. I can be in the lab. I will probably analyze a protein structure, and I will likely write a letter of intent because, one important grant deadline is coming up in September. I need to apply for a grant, right? So, not a single day is the same, ever. So, if you like routine, don't go to science.
Okay, also part of that. So what part of your whole research actually brings you excitement?
The most important one, the most gratifying moment in my career is when I see the ha-ha moment in my students' eyes. When they have a discovery and they made that discovery, and perhaps they don't realize the importance of that discovery, but I make them realize, what you just found out is pretty huge. Congratulations. So that sense of accomplishment in my students' eyes, for me, is one of the most important and gratifying things. To see my students succeed in their lifetime, pursuing a career, whatever career it is. I have former students who are scientists. They run their own research labs in Europe. I have former students that are working for pharmaceutical companies in the States. They make 3 times my salary. That's fine, right? But they accomplished their dreams. So to me, that's the most important thing that makes me go to work. They are my students, and I'm responsible for their successes. So to me, that's the most important, more gratifying moment. Obviously, when you get a grant, okay, that's fun, but that lasts for about 24 hours. After that, let's go to work, right? Or when you publish a paper, right? We publish a paper, the first thing I'm gonna say, it's a famous song by Jacques Brel, au suivant, to the next, right? So I say always the same thing. A paper is great, but this is one paper, right? Maybe this paper is important for one student, it's important for other people as well, but this is one piece of a very long journey that hopefully will get something at the end. So to me, I think the most gratifying moment is really when I see the success in my students, or my students being successful once they leave the lab. And obviously, I'm chair of the Department of Biochemistry, Microbiology, Immunology, and I feel exactly the same for the successes of my professors. When they get promoted, they get an award for their work, these kind of things. So for me, that's what makes me going.
Really interesting. Okay, so what keeps you curious in the scientific field? Like, what kind of piques your mind and gets you thinking, oh, this is really
I'm
interesting?
always on, seriously, even at night. So that thing doesn't turn off, and that thing is my brain, unfortunately. So there's not a single moment I say, okay, I had enough. Or I will always ask the next questions like, yeah, oh, that's great, but what? There's never no but what next. And so as a scientist, I think one of the primary criteria if you want to be successful in that career is be curious. Because if you think you've addressed all the questions, what's left to do, right? There's always a question. Science is never, it's never done, right? So we develop new technologies that allow us to do things that we were not able to do 15 years ago. So what we thought we knew back 15 years ago has to be revisited because now the model has changed, the technology has improved, right? So for over the last 30 years or 45 years, the amount of technology that's been developed is just mind-blowing. And it's just allow you to do more. And get even more insights into a biological process and or the state of a disease, for example. So yeah.
And your approach to science is like a mix of creativity and science. So is there like maybe like a personal story or like experience that actually like shaped how you approached it?
I think the story in Cégep kind of shaped up who I am. And I never said thank you to these two professors. One was the organic chemistry Professor Girard at Cégep that would let us go in the lab, and he was just providing advice and say, "What?" And he was telling us like, "You guys, you do what you want to do. You know, like I'm just here to help." And the other prof was teaching a microbiology course in which he had given us the task to isolate in a mixture of 3 bacteria. We had to isolate each bacteria individually to create pure culture. Okay. But he said, "Figure it out." So to me, it was like, okay, I need to read the literature. I need to figure out what this, and he told us like, okay, this is part of this genera, this part of this species. And so it gets you some sense of where you need to look. But really the challenges, the curiosity, the finding out as how, and then the reward at the end when you succeed. And the thing is that what I hate the most, but what I like the most is when I fail. But then I do it again, and I do it again, and I do it again until it works, right? So never giving up. So one second important characteristic you have to have as a scientist, stubborn / never giving up, right? It's because 95% of the experiments that you do are failure. 5% of them will work. But that you'll be really happy about the 5% that will make you forget the 95%.
Yeah, well, it must be really rewarding to like overcome those challenges in the lab.
Yeah, and you always learn something, and even in your failure, you learn something, right? You learn that when you do this way, it doesn't work. So let's not do it again, right? Mm-hmm. So there was this famous quote by Edison saying, "Oh, you got to try how many times to build a light bulb," and he succeeded in like I don't know how many times, and every time was saying, "No, I didn't fail. I just found out how not to make a light bulb."
Yes,
right.
Exactly.
But this is exactly the same thing, right? And in science, it's all about that. We always joke around. With my students, if there was some sort of journal of failed experiments, they would get a lot of papers because everybody would say, we tried that, no, don't do it, it doesn't work. And we tried that, no, don't do it, it didn't work. And I guess it would save a lot of money. But yeah, I mean, this is part of science. And when you're curious and you want to really achieve your goal, then it doesn't matter that you fail 95% of the time. And just give you an example, in my lab, it took literally 13 years to solve one protein structure. And it's just, we didn't figure it out how to do it. And I was on sabbatical this year to learn a new technique in Grenoble. And I had actually, in my network, going back to the network and importance of it. So I was in an institute that there was tons of structural biologists. Mm-hmm. And I started asking questions, I'm really having issue with that, how to solve this structure. And one guy was like, well, you should do this, do that, and I think you give it a shot like this, and I think you're gonna nail it. I didn't know how to do that. I tried it, and it worked, right? So it took 5-6 years to actually get the sample ready and purified, wow. And then 6 years to get crystals or to get the samples to expose the X-rays with, and it took us another 3 years to actually solve the 3D structure. Now, I don't want to scare you. It's not always that long, okay? So I still want to recruit grad students in the lab, right? But yeah, that's an example is that we failed for so many years, but when we nailed it, when we got it at the end, it was like, wow. And nobody has been able to crack that structure, and there's a reason for it. It's because it's really hard. You have to know how, and the different things that you need to do, and it worked out at the end. But So to reassure you, no grad students was working on that project. Okay, it was only research technicians and professionals were working on it.
All right, so now we're going to kind of switch over to looking at what you think the future holds. So, in 10 to 20 years, how do you think your field will change, or how do you hope it's going to change?
Yeah, that's a good question. I think AI will have a massive impact. It already does to some extent. I mean, the publication of the third iteration of a program called AlphaFold. Not sure if you heard about that. As a fourth-year students, you definitely did.
Yes.
As a second-year students, maybe not.
Yeah, would you mind breaking that down a little bit just for listeners in case they haven't heard
so
of it?
AlphaFold is a machine learning algorithm that actually built on different, would say, pipelines, for lack of a better word. And essentially, it used a lot of different informations that some of them were already out there to some extent. Some of them have been developed. And really, what it does, from a primary amino acid sequence, they will provide a 3D prediction of your protein. So it's been a long process in the making. There were great documentary on this. For example, there was a competition for many years called CASP, and they were trying to build algorithm or tools to actually go from the amino acids sequence to the 3D structure. And the prediction tools were doing a good job, but they were kind of stuck. And at some point, they started, I guess, Google and OpenMind got into the business, and things accelerated to some extent after that until they created the first version of AlphaFold. So AlphaFold is really about that. So you can go online. You only need a Google account, seriously, and you go there. You take the favorite amino acid sequence of your protein, put it in there, and that will generate a 3D model.
Oh, wow.
Oh yeah, and so and for several proteins, it does a very good job compared to what was out there before. So that's a massive improvement. And so, yeah, I think this has been a major breakthrough. So there's obviously sometimes the program will make some mistakes. And as a structural biologist, we're there sometimes to look and see, okay, maybe it makes sense or maybe it doesn't make sense, right? So, but that was a major breakthrough. And I think it's going to get only better in a sense that now we're going to actually, and it started already, now we have protein on their own, but obviously most of the protein or several proteins work as part of protein complexes, right? So now the next generation will be, okay, how we model these protein-protein interaction or these 4, 5, 6, or 7 subunit complexes and how they assemble together. So that's really, I think, in the pipeline. Also, improving in silico methods for drug docking or for small molecule docking on predicted protein structure. So I think there's a strong appetite toward that. So I think on the structural biology level, I think this is really where the field's going to go. And also this is a relatively old technique, I would say, but it started to be to gain interest a lot in the last 15 years or so, called cryo-electron microscopy, which in cryo-EM that allows you to get the structure of a protein, but but for cryo-EM, the people use cryo predominantly for when they can't get those crystals or using X-ray crystallography, and typically those are from big protein complexes because they're hard to crystallize. So, and the technology is a little bit different. The physics behind is a tad different, obviously, but at the end of the day, you get the 3D images of your protein, and similar to X-ray crystallography, you can extract similar type of information. Now, the technology has improved quite tremendously on that from in the last 15 years. Just detectors, the softwares we use to analyze the data, but there's more and more that's come in the future to improve, again, the amount of details you can get out of these structures. And in combination, and obviously when you have AlphaFold, that is part of that pipeline with cryo-EM structure, that feeds in very positively together to determine at very high resolution large macromolecular complexes. So I think this is where the future will be in the next 15 years, actually.
All right, so moving on towards gearing it towards the scientists of the future and some of the students that we're going to be talking to and are going to listen to this podcast. So what's some of the advice that you have for these future scientists and students that are interested?
It's a good question. That's a great question. I think what I found in my career, there are several important things that a student need to be proactive on really early on. And one of them, and I mentioned it, is networking. Networking, because the students that you are studying with will likely, a fraction of them, will become actual colleagues or doing something similar in your life. So already at the undergrad level, don't ignore the importance of your network, okay? Don't suffer from the imposter syndrome. Don't, ever, okay? If you want to do something, you can do it, okay? It's going to require sometimes an higher level of motivation, okay? Don't have the imposter syndrome. So as an undergrad, you don't have this opportunity yet. Well, maybe you do to some extent. Let's say you want to chat science with a prof, but you're too scared. Don't be. They will not answer your email if the email is uninteresting to read. Okay, what I mean by that is if you really want to chat science with this professor, you better have read the papers that he has or she has written and arrive with specific questions, because so at that level already, don't have the imposter syndrome. Don't think that you will understand the papers completely. You probably get maybe 25% of the paper, but at least you tried. And that shows a lot, okay? So that's an advice. No imposter syndrome, build your network, don't be afraid, okay? So, and when you do this, sometimes that opens door that you didn't anticipate. So if you arrive and you meet a professor without perhaps even the intent of joining the lab, okay, but you want to discuss science because you read this and for some personal reason you're interested in this, okay, go. But you better be prepared. Read the papers, be professional about this, okay? But, a few years later, you're looking for graduate studies or a spot for graduate studies. You're not necessarily interested in that PI's research, okay, or that professor's research program. That's perfectly fine, right? There are tons out there, and that might not be aligned with your research interests anyway. But that person will remember you. And the way you approach the discussion, the professionalism that you had when you meet that professor, and that will not be forgotten. What that gives you? A reference letter. What that gives you is that you give that person's name, that professor's name, to the one that you want to reach, and you can be sure 100% that when that happens, a student applying to my lab and provide the name of a prof as, potential or, providing some sort of assessment, I will reach out to that person, okay? So that's one particular example. When you go to graduate studies, you continue. If you already started that at the undergrad level, you're going to be in the groove and you just continue doing it. And that applies when you go to conferences. One amazing example that I give my former PhD student, Pam. Pam was absolutely phenomenal PhD student, published 5 or 6 papers in the lab. Really highly productive PhD student. But one thing that Pam had, she didn't have imposter syndrome, and she was not shy of approaching what we call the outstanding scientists in our community of epigenetics. And she was actually taking by hand a prof and say, come to see my poster, I want to show you. And you know what? None of them said no. They went because they were legitimately interested. And they were seeing a motivated PhD student, PhD candidate, wanting to show and explain the science. No imposter syndrome, not being afraid to take the first steps, engage in discussion, build your network again, right? And that continues, right? So that's the first, this is an important really aspect of your work, and you will never network enough, okay? Because at some point your scientific career will be built or will be tremendously helped with that network. So for example, if I didn't have great collaborators at Yale University, I would have never published a science paper 2 years ago. Same thing about 7 years ago, right? It's because I have these collaborators that we share, that we have different expertises, that we can push better science together. Okay, so network. Communication is key. Gain confidence. Be curious. Be stubborn, but listen as well. Open to advice, right? You can be stubborn, but if you repeat the same mistake 15 times, okay, there's another word for that, but I ain't gonna say it, right? But you have an advisor, you have a mentor that is there to really guide you. So listen, right? Again, no imposter syndrome. You can do it. I get a lot of that, I get a lot of these students like, I don't want to become a PI, or I don't want to become a scientist. Why? Well, because it's hard. Really? I mean, you like doing science? Yeah. You like answering questions? Yes. You like to be in the lab? Yes. You're really good at writing science? I can be better, but we're never perfect. But we're pretty good. What's not to like? You're in it. It's just, this is the imposter syndrome talking. Oh, I can't do it. I can possibly do it. It's too hard. No. I had that syndrome, but I decided to continue anyway.
Yeah, so it's really like about being confident in yourself and your abilities, I guess, is what you're trying to get across.
Yeah.
And to like have no fear when approaching people, which I think is something that a lot of people do struggle with.
I know a lot of people too that like suffer with imposter syndrome. Like it's very,
I know,
very
a
common.
prof is a human being first thing, right? So we listen, and we've been through what you've gone through a few years back.
I think a lot of us just forget about that piece.
Yeah.
All right, and then our final question for you today. So do you have any opportunities or ideas of opportunities for students who want to get engaged in your field?
There's always opportunities for students who want to have opportunities. I gotta simply say that. So if we'll never say no to a motivated student. Again, you need to show the motivation, and something that we realize in BMI, and my dear colleague, Dr. Emilio Alarcon, develop a guide for students, and it's on the website actually, on how you should approach a prof if you're interested in an opportunity, right? And what we realize is the students don't know what to do. They don't know how to engage, and we get that same generic email, oh, I got a good A+, or I got a good CGPA, I'm interested in, for example, your science, without saying what kind of science he or she's interested in, and really kind of a generic email. I will tell you right off the bat, even for the most motivated student, writing just that bits of email, the email gets in the trash really easily. So there's always opportunity, always, right? But I will perhaps narrow my statement in saying there's always opportunities for those who really want to have an opportunity, right? Because I get 50 emails, I said 50, not 15, 50 emails per day of students wanting to join my lab. It's always the same thing, always the same mistake. And the result is all the same, go to the trash. I delete. I read them in 30 seconds, and because they're not interested, they're just trying to find a place, to find a place, right? But if you think about this, let's say you apply for, a big time job. Are you just sending like a generic email, so, hey, I'm interested in your, advertisement, No, you're gonna search a company, what are the core values? where they're going. There's probably a strat plan available. What is their biggest achievement? What is their, if it's a company, if it's a lab, what are their latest publications? You go on different websites, whether they have CIHR or NSERC money. So those are typically grants that we get here for researchers in the Department of Biochemistry, Microbiology, Immunology. You see what are the projects. Then you can refine your email tremendously and say, hey, I read this, I read that, and I'm really interested. Have you thought about these questions? Just this. So, and it means that if you have questions, it means that you've read some aspects of some papers. And if you read a paper, it's always, the formula is always the same. At least we teach that in TMM, how a scientific paper is packaged. There's always few outstanding questions left off in the discussion, for example. You can start off with that, right? So again, you have to make your opportunities by networking, but don't think that just, even though you have a CGPA of 10, that just sending an email, a generic email to a PI will get you an interview, okay? You need to have students, because when you take a student, this is an investment, right? And you're investing in that student a lot on their future. So we're not taking any students. We're taking students that are motivated by research, right?
Yeah, you want students who are actually interested.
Interested in what I'm doing, or what somebody else is doing, right? So I don't mind, and also be open, right? Don't make the mistake of narrowing unless you really know what you want to do. Don't make the mistake of narrowing one lab, right? For example, you can be interested, let's say you're interested in Parkinson's diseases, for example, okay? I'm going to give you an example. There are several outstanding laboratories here in RGN and at OHRI that are working on Parkinson's diseases. They use different tools, they use different techniques, so you learn different aspects on how to study Parkinson's disease. So if you're interested in that and you're really working, and reading, making an effort to read papers or few papers of one lab or another that are working on Parkinson's disease and using different techniques, you apply, and they both want to meet with both of them. And don't be afraid to ask, what is the lab philosophy? What are the opportunities to network? What are the opportunities to present at conferences? Because again, all this as a grad student, because all this actually helps you push your scientific career, right? So for example, I send my students undergrad, fourth year undergrad, when there's a good timing, I send them to conferences. So they present a poster. So again, if you apply for a scholarship, for example, well, if you presented a poster at a conference, let alone an international conference, that is worth a lot, actually, right? I mean, obviously, an undergrad student, sometimes they may not have time to have their name on a paper. Sometimes they do, actually, depending on the nature of the work. But you have to ask these questions. So, and when you interviews with both, or one after the other, you say, well, okay, thank you for your time. Really like the lab, but you can be open about this. Like, I'm also interviewing with Dr. I'm also interested in, and typically the PI will be okay with that because you explore, this is your future, right? So this is important to explore, and you are at a stage in your career that you can do this.
Thanks so much for joining us, Dr. Couture. We really appreciate discussing with you how exploring chromatin structure and epigenetics brings together science and creativity. And thanks to our listeners.
I'm Anusha.
And I'm Amelia.
And we'll be back next time with more stories, insights, and a fresh cup of tea to spill. Remember, the journey doesn't stop here. Keep thinking, keep questioning, and keep steeping in inspiration. See you next time.